Hello, good evening. Welcome to the Experimental Biochemistry Week 9 live session. I am Surasree. I am a Prime Minister's Research Fellow working in the Department of Biotechnology, IIT Kharagpur. In this experimental biochemistry course, we have in our previous sessions we have discussed about the structure of protein, the structure of amino acids, how to isolate protein, purification of proteins, how to perform the spectroscopic techniques, and fluorescence spectroscopy, absorption spectroscopy, things like that. In this session, we are going to discuss about isolation of DNA, isolation of plasmid DNA as well as isolation of genomic DNA. So before going into that, let's begin the session with the discussion about central dogma. Now, what is central dogma? So, central dogma actually is the principle that runs central dogma is the dogma or the principle that is followed in all life forms, and it is the principal theory of the molecular biology. So it states that these three biomacromolecules, the DNA, the RNA and the protein, how they can be formed, how they can be generated, from which by which process they can be generated. So first let's talk about DNA. So DNA is, we know that it is the carrier of genetic information. So how does DNA increase in number in the cell? Or DNA, we know that it is the since it is the carrier of genetic information, stretches of DNA make up the genes, right, which carry the several sequences. Genes are the sequences that carry the information for coding the proteins, right? So the information that we carry from generation to generation lies in the DNA. It is given by our parents. So we receive our DNA from our parents, right? Now this DNA carries all the information of life. Now all the information of life, what do I mean by that? So basically our life forms in our cells, if you consider a cell, the molecules which are functional molecules, those are actually proteins, except for the RNA. But I'm going to come back to RNA again. But the functional molecules, the molecules which carry out maximum function structurally and functionally, they are the protein molecules. Now DNA is the carrier of genetic information and proteins are the functional units. So now how is this how is the bridge joined between the DNA and the proteins? So central dogma is actually the theory about that, how the information is transmitted from DNA to protein. So scientists have discovered that there is an intermediary molecule in this signal transduction pathway, so or in this information transduction pathway. So the intermediary molecule is actually RNA. So DNA, first of all, DNA can copy itself. So which means that DNA by itself can increase in numbers. What is this process called? This process is called replication. So by the process of replication, DNA can increase in number and it can make a copy of itself, an exact copy of itself. Now DNA, the information in the DNA is carried in the nucleotides. What are nucleotides? I am going to come later. Now this information that is held in the DNA, if it needs to go to the proteins, proteins are, we know protein structure, we have studied already that proteins are made up of amino acids. Now the nucleotides, from the information that is present in the nucleotides, how does this information gets transmitted to the proteins which are made up of amino acids, since these two are completely different biomolecules. So the messenger here is the RNA. So RNA or ribonucleic acid carries the same information as the DNA in the nucleotides. So from the DNA to the production of RNA, this process is called transcription. So as if making a transcript. So you know what is a transcript, right? It is a copy of something. So DNA copies its genetic information by the process called transcription to form RNA, or very specifically messenger RNA, which now carries the information for protein synthesis, right? So this is the messenger molecule, which is why it is called the messenger RNA. There are other types of RNA as well, transfer RNA, ribosomal RNA, but the information carried from DNA to protein, that function is done by the messenger RNA or mRNA. Now this mRNA carries the information that was previously the DNA. Now it is in the RNA. Now this RNA will translate this information to form the proteins, the functional molecules of the cell. Now this process is called translation. So it is the process of protein synthesis from the mRNA sequence. Now the mRNA or the RNA molecule, it is also made up of the same, more or less same structure as that of DNA. We are going to discuss later on what is the difference between DNA and RNA. But the RNA contains the same units, that is the nucleotides, as that of DNA. Now with the help of a complex machine which is called a ribosome, this information that is there in the RNA is translated to form the proteins. That is the theory of the central dogma. Now let's look at in details the structure of DNA. So before going to the structure of DNA, let's first discuss the structure of nucleotide. So nucleotide is what? It is the structural unit of nucleic acids, or the monomer, right? That is the nucleotide. Now how does this look like? What is the structure of this nucleotide? So this part of the nucleotide is called the deoxyribose sugar and nitrogenous base. So nucleotides are actually formed of a nitrogenous base joined to a ribose sugar plus phosphate moiety. So these nitrogenous bases, they are of several types. They are classified into two groups, these nitrogenous bases: purines and pyrimidines. Now purines, this is the structure of the purines. The purines are adenine and guanine, while the pyrimidines are cytosine, uracil and thymine. Now the purines, you can see that these molecules have two rings, and the pyrimidines, they have only one ring structure. Now the purines are very similar in structure, while the pyrimidines are also very similar in structure. That is why they are classified into these two groups, purines and pyrimidines. Now you have to remember that the difference between DNA and RNA, one difference between DNA and RNA is that in case of pyrimidines, cytosine and thymine, they are present in DNA, while cytosine and uracil, these are present in the RNA. So basically, DNA has these two pyrimidine bases, C and T, while RNA has these two pyrimidine bases, C and U. So thymine is replaced by uracil in case of RNA. Thymine is replaced by uracil. And remember, this you should pronounce as thymine and not thiamine or thiamine. That is a different molecule. That's a vitamin actually. So this is thymine. Now this thymine is present in only DNA, whereas this uracil is present in only RNA. And you can see the difference in structure between them. The thymine only contains an extra methyl group than the uracil. Now these are the nitrogenous bases. Why are they called nitrogenous bases? Because there are several nitrogen atoms, right? Now there is this red line shown here, right? What does this stand for? This is actually the nitrogen molecule through which it is connected to the ribose sugar. So the nitrogenous base is connected by a glycosidic linkage to the ribose sugar. Now this ribose sugar is also different in DNA and RNA. In DNA, it is deoxyribose. That is why it is called deoxyribonucleic acid. And in RNA, it is ribose. That is why it is called ribonucleic acid. Right? So now this nitrogenous base is joined to the or attached to the ribose sugar by this glycosidic linkage. And this together form the nucleoside. This is called the nucleoside. So you can see it here, the nitrogenous base along with the ribose sugar. Now when a phosphate group is attached to this nucleoside, it is called a nucleotide. So that is why these three together, it is called the nucleotide. So this one is the nucleotide, the nucleoside monophosphate, that is basically the nucleotide. So you can see that this phosphate moiety is attached to the 5' hydroxyl group of the ribose sugar. Right? This phosphate molecule is attached with this 5' hydroxyl group of the ribose sugar. Now this structure can actually be formed up to 3 phosphate moiety, with up to 3 phosphate moieties. So when second phosphate is attached, it is called the nucleotide diphosphate, and the third phosphate is attached, it is called the nucleotide triphosphate. So it is called nucleotide triphosphate or NTP. Okay. Now here you can see the difference between the ribose and the deoxyribose. So the difference is in this 2' hydroxyl group. So in the 2' carbon, you can see in the ribose sugar there is hydroxyl, but in the deoxyribose, in this 2' carbon, there is only hydrogen. So this oxygen is missing, that is why it is called deoxyribose. And this is present in DNA and ribose is present in RNA. And we will see later on that due to this presence of the hydroxyl group in the ribose sugar, RNA is less stable than DNA. So far any doubt about the structure of nucleotides and the components of the nucleotide? Okay, so let's proceed if there is no doubt. So now we will see how these nucleotides are joined together. So I told you that these nucleotides are the monomer and the polymer is the nucleic acid, right? So how are these monomers joined together? So they are joined by phosphodiester linkage. So you can see this is the structure of a DNA molecule, but only a single strand of the DNA molecule. So this is a single strand is shown here. Usually DNA has 2 strands and they are helically wound around each other, but this is actually showing only one strand of DNA. And here you can see the nucleotide structure. So this one is your nitrogenous base, this is your ribose sugar, and this is the phosphate group. Now DNA molecules, they are usually they have 2 terminal. One is called a 5' terminal, another is 3' terminal. So how are these 5' and 3' determined? You can see that in this beginning, in the first nucleotide, that is in this first nucleotide, you can see there is this, at the end there is this 5' phosphate, right? The phosphate is attached to the 5' hydroxyl group. So that is why this end is termed as the 5' end, while if this is suppose the last nucleotide, you can see that there is a 3' hydroxyl group at this end. So this is why it is called a 3' end. So DNA strand always runs from 5' end to 3' end. And now let's look at how are these nucleotides joined to each other. So this 3' hydroxyl group, which was present here, now it joins to the 5' phosphate group of the next nucleotide. Okay, so this is the 5', this is the 4', this is 3', 2', and this is 1'. Okay, now this 2' hydroxyl is present only in RNA. This 3' hydroxyl and this phosphate, which will be attached to this 5' end, they are joined in consecutive nucleotides, and this linkage is the phosphodiester linkage. So here you can see this, that the 5' phosphate of this nucleotide 2 joins with the 3' hydroxyl of the nucleotide 1 and forms this phosphodiester linkage. So there are 2 ester linkages here. That is why it is called a phosphodiester, and thus these nucleotides, they form the chain. So this is the second nucleotide, this is the third nucleotide, this is the fourth nucleotide, like that. So now like this the chain lengthens, right? So here you can see that the nitrogenous bases, they are like adenine, guanine, cytosine, thymine. These nitrogenous bases are present, which link to the ribose sugar, and the DNA chain is joined by the phosphodiester linkage. Now the next thing is the structure of the DNA. How does the nucleic acid look like? So I told you that this DNA forms this double helical structure. Which was discovered by, or first solved by, Watson and Crick, the double helical structure of DNA, which was observed in the crystallographic image. Now this double helical DNA structure, it forms these grooves kind of things, minor grooves and the major grooves, and they are these strands, the two strands that form the double helix, they are antiparallel. The two strands are antiparallel. So one runs from 5' to 3' and another runs from 3' to 5'. And the bases, so the backbone is actually, the backbone of DNA is formed by the ribose sugar, while the bases, they protrude inwardly and they form pairs with each other. So how do they form pairs? Mostly by hydrogen bonding, right? So these purines and the pyrimidines, they can form hydrogen bonds between each other, but there is a specificity in that. That is, thymine, here you can see thymine can form two hydrogen bonds, 1 and 2, two hydrogen bonds with adenine. So thymine can form two hydrogen bonds with adenine, and cytosine can form 3 hydrogen bonds with guanine. So thymine and adenine, they form 2 hydrogen bonds, and guanine and cytosine, they form 3 hydrogen bonds. So these bases are protrude inwardsly, protruded inwardsly, while the backbone of the DNA is formed by the ribose sugar, right? And that is how they form this pairing by the hydrogen bond is actually the basis for formation of such double helix structure. So these base pairs, you can see here, these base pairs are present in between the two strands of the DNA. Now the important thing is, DNA exists in this double helical form but not RNA. Why? Because RNA is a single stranded molecule. So it contains only one strand, unlike the two strands of DNA. So if we look at the structural difference between DNA and RNA, we know that DNA contains one strand, DNA contains two strands, while RNA contains only one strand. DNA contains deoxyribose sugar, while RNA contains ribose sugar. DNA has these bases A, T, G and C, whereas T is replaced by U in RNA. So these are the basic differences between DNA and RNA. Now this difference of the ribose sugar also makes DNA much more stable than RNA. So how is that? If you remember, in the last week we were discussing about the mechanism of action of RNAs, enzyme RNAs. Now in this mechanism of action we had discussed how this RNase molecule, it has two histidine in the active sites, in its active site, and these two histidine molecules, they can act as an acid or as a base to destabilize or to break the phosphodiester linkage of an RNA molecule. Now for the molecule which is the group which is responsible for this reaction is this 2' hydroxyl group. So this is the structure of RNA. That is why you can see this 2' hydroxyl group. If it was DNA, there wouldn't be any hydroxyl group, there will be only a hydrogen atom in the 2' position. So now if you add any base, then what will happen? This base will attack this hydrogen, the hydroxyl group. Now it will extract this proton out. Therefore there will be a bond formation between this oxygen and the phosphate group, and simultaneously this particular bond, this bond will break. So now you can see that by this action of a base or by the action of this enzyme RNase, the nucleotide 1 and nucleotide 2 will dissociate from each other. So the phosphodiester bond will be broken. This phosphodiester bond will be broken. So this particular bond that was linking the two nucleotide molecules, this particular bond will be broken, right? And the two nucleotides will be separated from each other, right? So basically the RNA chain will break. Now this kind of reaction does not occur for DNA because DNA does not have this hydroxyl group, so it is not prone to this kind of an attack, right? So this is the reason why DNA is much more stable than RNA, which is why it is assumed that DNA carries the genetic information and DNA evolved as the carrier of genetic information in the course of evolution. So because RNA is a molecule which not only carries genetic information but also it acts as an enzyme. So it has the properties of nucleic acid as well as it has the properties of protein. So many scientists assumed that RNA is the molecule which began life, or which is actually the molecule which is responsible for the evolution of life. So that is called the RNA evolved hypothesis. But the contrasting theory to that is that RNA cannot be the first molecule of life because RNA is not as stable as DNA. So that is one theory. Now after discussing this structure of RNA and DNA and nucleotides, now we will move on to the isolation of DNA. And what do you mean by isolation of DNA? So where from DNA can be isolated? So what is the source of DNA? So ideally from any cell you can isolate DNA. But in laboratory we isolate DNA from specific animal tissues or bacteria or plant tissues. um, and also DNA can be isolated from like archaeological remains also, like something that is very old, from that also DNA can be isolated, which has been fossilized animals, for example, or fossilized plants. From there also you can isolate DNA, but the process is difficult, but DNA remains stable in that condition. So DNA is a very stable molecule. Now in laboratory, we can use animal tissues like liver, spleen or kidney for isolating DNA from these animal tissues. Now in bacteria, so bacteria actually has two types of nucleic acids. One is a genomic DNA, another is a plasmid DNA. So what is the difference between a genomic DNA and a plasmid DNA? So the genomic DNA is this long DNA molecule or the chromosome of bacteria that carries the genetic information, or that carries the information to be transferred from one generation to the other, and it carries all the information for the life cycle of bacteria. But there is these circular molecules also, which are called the plasmids. These circular molecules of DNA, they are also important to the bacteria because they confer several characteristics to the bacteria. For example, this antibiotic resistance. So often we talk about this phenomena, that is the resistance of antibiotics in the bacteria. That is like we are suppose having one particular antibiotic, and we see after several days that the disease is not actually subsiding, or the bacteria can actually multiply in presence of this antibiotic. So which means the bacteria has become resistant to the antibiotic. Now this antibiotic resistance gene is one such trait which is carried in the plasmids of the bacteria. Now there are other traits also. For example, this conjugation, for the conjugation there can be several plasmids, the F plasmids and all. So there are several traits that are carried. So these are also DNA molecules, but these are circular DNA molecules. And the difference between the genomic DNA and the plasmid DNA is that genomic DNA is much more long, while the plasmid DNA is very small. So approximately the plasmid DNA can be of 10 kilobases. For example, the plasmid, if it has like, for example, 10 kilobases, the genomic DNA is much longer than that. So genomic DNA is approximately 5 into 10 to the power 6 bases, right? So 5 MB, that is the genome size, while the plasmid size ranges in kilobases or KB. Now that is the bacterial DNA. So from bacteria we can isolate plasmid DNA, we can separately isolate the bacterial chromosome or the genomic DNA as well. Now from the plant, ideally from any plant part we can isolate DNA, but there is a problem in the plants, that is the plants have a tough cell wall. The plant cells have a tough cell wall. And not only that, there is polysaccharides as well as the secondary metabolites present in the plant cells, so which actually makes this process little difficult to isolate DNA from the plant cells, but it is possible. So these are the sources of our DNA. Why do we isolate it? For many downstream processes. So suppose we want to clone a particular gene and we want to express the protein. So for cloning that particular gene, we can clone it in the plasmid DNA, which we isolate from a bacteria. We can take out this plasmid DNA, that means we can isolate this plasmid DNA, and then we can do some manipulations in the plasmid DNA and then put it again back to the bacteria, right? So these manipulation will be, suppose for example, cloning or any mutation, Now again, if we put it back in this bacteria will grow this plasmid DNA. It will make several copies of this plasmid DNA, and suppose if we have cloned our particular gene of interest, it will express the protein, right? So these are several things that can be done. Cloning, manipulation of the DNA and things like that. Now, what is the method of this plasmid DNA isolation? So first we will talk about this plasmid DNA isolation, and then we will move on to the isolation of genomic DNA. So for plasmid DNA isolation, the method is called the alkaline lysis method. So before going to that, let's see how we can grow the bacteria from which we will isolate the plasmid. So that will be that step 1, growing the bacteria. So suppose this is a plate where this is an agar plate, right? This is an agar plate where there are several colonies which are formed. So we have grown our bacteria on this agar plate and it has formed these colonies. So now what we will do is we will use a sterile toothpick to pick one colony from this plate and then inoculate it in a culture medium. So this culture medium is LB, Luria botany medium, and we will add specific antibiotics to it if the bacteria contains some antibiotic resistance plasmid, right? So usually what is done in our lab, the plasmids with which we work, they are manipulated or they are made recombinant with the expression of various antibiotic resistance genes. So that is why to grow that particular plasmid, we put the bacteria under antibiotic stress. So this antibiotic can be kanamycin, for example. It can be chloramphenicol. It can be ampicillin. So depending on the antibiotic resistance gene that is present on our plasmid. So this antibiotic resistance gene, if suppose our plasmid contains this antibiotic resistance gene of kanamycin, then what it will do is that it will produce resistance against kanamycin bacteria, against kanamycin antibiotic. So if we grow the bacteria in presence of this kanamycin, the bacteria will be able to grow, right? Because the plasmid will contain the kanamycin resistance gene, so it will provide resistance to the bacteria against this antibiotic kanamycin. Now this kanamycin, the presence of or the stress of this antibiotic kanamycin will also select the bacteria which have this plasmid against the bacteria which does not have this plasmid, right? So the bacteria which will not have this plasmid will not grow in such a medium, because it will die because of this action of the antibiotic. The bacteria which do not have this plasmid, but the bacteria which will have this plasmid will grow in such a medium. And so what we will do is we will allow overnight growth at 37 degrees centigrade incubator. So we will take an incubator and we will put this culture inside the incubator and we will also shake it. So the incubator will actually shake in a particular RPM, so that there is proper aeration and the growth can be better, right? So we will keep it overnight at 37 degrees centigrade and we will allow this bacteria to grow. Now next morning when we go to the lab, we will take out this culture and then we will transfer it into a tube, in a centrifuge tube, and then we will centrifuge the culture. So if we centrifuge the culture, for example at 6000 rpm for example, then what will happen, the culture, from the culture, the cell debris, not the debris, the cells, they will form a precipitate. So the cells will form a pellet, and the rest of the media which do not have any cells, it will form the soup or the supernatant, and the supernatant can be discarded. So now, starting from the bacterial culture on the agar plate, we have now got the cell pellet, and we are assuming that this cell pellet will contain our plasmid of interest, because this bacteria has grown in this antibiotic, in the presence of this antibiotic, so it must have the antibiotic resistance cassette. So this is the cell pellet that will form. Now the next step will be to lyse the cell. The plasmid DNA will be inside the cells, right? Now if we do not lyse the cell, then the DNA will not be able to come out in the solution, right? So that is why we will lyse the cell, and that is called the alkaline lysis method. Why alkaline? Because it is conducted in a alkaline medium. So now the next step is, we will first resuspend the pellet in a resuspension solution. So we will first resuspend the pellet in a resuspension solution. So resuspend means what? So it means the pellet, you dissolve the pellet. That is called resuspension. So you use a particular solution to resuspend the pellet. The next step is alkaline lysis. That is, you now lyse the cell. What are the components in this alkaline lysis solution? We will discuss later on. And the next step will be to neutralize the solution. That will be neutralization. Followed by this lysis and neutralization, next what we will do is that the supernatant that will be collected, the supernatant, so we will actually centrifuge at a very high speed, and then we will get a supernatant. Now the supernatant is actually the solution that will contain this plasmid DNA. So now this supernatant we will load onto a column. So this is a column. Sorry. This is a column, and on top of this there is a membrane. This is actually a silica membrane, and we will put this solution in this column. We will put this supernatant in this column, and we will allow the DNA which is present in the supernatant to bind to the silica membrane of the column. So first we will allow the binding of the DNA to the silica membrane, then we will wash the membrane with some buffer, with some ethanol, a solution containing high ethanol percentage, and then we will finally elute the plasmid DNA from this membrane, right? So the DNA now has bound to the membrane. Now we will apply either water or tea buffer to elute out the plasmid DNA from the membrane, and now this water or tea buffer solution will contain our plasmid. So that is the overall step of plasmid DNA isolation by the alkaline lysis method. Now I will go in details about this method. So first step is alkaline. After you resuspend the pellet with a resuspension solution, The next step is alkaline lysis. So for alkaline lysis, the first thing is bacterial cells, they have cell walls, right? So for lysing the cells, we use lysozyme. Lysozyme can be used. Next thing is, this is alkaline lysis, so we will make the medium alkaline. Why? Because the alkaline medium will reduce the interaction between the DNA and histone proteins, right? Then we will also add, so in this alkaline solution we will add NaOH and SDS. NaOH, sodium hydroxide, will make the solution alkaline, and it will precipitate the DNA and the proteins. We will also add EDTA in this solution, because EDTA is a chelating agent. So EDTA will chelate these divalent positive ions, the divalent positive ions, the magnesium, manganese, these ions. And what is the role of this chelation? What is the function? The DNAs, these enzymes, they need this cofactor magnesium. Magnesium ion is used as a cofactor for these enzymes. So if the EDTA can chelate these magnesium ions, chelate means they can bind to these magnesium ions, so the magnesium ions will become unavailable to the DNase enzyme, and therefore the DNase enzyme will become inactive. So why is it important? So that DNase cannot chop off our plasmid DNA of interest, right? So that is why DNases are made inactive by the action of EDTA, and also the cell wall also gets destabilized because of the chelation of the manganese ions. Now there is these two components, NaOH and SDS. Now SDS is important because SDS is a surfactant, so sodium dodecyl sulfate. This actually solubilizes the cell membrane, the lipids of the cell membrane, and it also helps in denaturing the proteins. We have seen that when we perform SDS page, SDS is used to denature the proteins, and SDS also disrupts the protein DNA complexes that can form. And we also keep high salt concentration to diminish the protein DNA interaction. The action of NaOH is to precipitate the DNA and the proteins, and also we use RNAs in this lysis step. RNAs we mix actually in this lysis step. Why? Because RNAs is used to remove any RNA contamination. So DNA and RNA, we know that they are both nucleic acids, right? So when we will try to isolate DNA, RNA contamination may also come, and that will that will actually affect the quality of the DNA isolated. So to remove this RNA contamination, we use this enzyme RNase. So that is the step of lysis. Now next step will be to remove the cellular debris. So first we will neutralize the solution, and then what we will do is that for neutralization we will use an acidic solution. So previously our solution was alkaline. Now for neutralization we will use this acidic solution of potassium acetate. Now when we apply potassium acetate, it will precipitate the dissolved proteins and the membrane debris. So by the action of this acidic solution of potassium acetate. Now also potassium will form a potassium salt of this dodecyl sulfate, which becomes insoluble and it will precipitate. Now in this particular step, for removal of the cellular debris from our solution, so now in the previous step what happened, we have lysed the cell. right? So now once the cell is lysed, all the components of the cell will be out in the solution. Now from that we need to isolate our DNA, or isolate the DNA from the plasmid DNA from this population, mixed population of many macromolecules and the cellular debris as well. So now how do we remove these cellular debris? First we neutralize the solution with potassium acetate. Now next what we do, we centrifuge the solution. So centrifugation is done at high speed, and now in this step what will happen, the large size of genomic DNA will actually prevent it from annealing. So the DNA and the proteins which were denatured in the previous step, right, the DNA was denatured because of the action of NaOH. DNA will get denatured because of this action of NaOH. Now this denatured DNA, it has to anneal. Now the genomic DNA of bacteria, it will have large size. So due to this large size, this genomic DNA will not be able to anneal, and therefore it will precipitate. However, the small plasmid DNAs are much smaller than the genomic DNA. So it will quickly anneal. So the two strands will quickly anneal, form base pairs, and they will remain dissolved during this step. So what we are seeing here, that all the debris, the proteins, the genomic DNA, these proteins, the lipids, the genomic DNA, everything is coming into the precipitate. So only after centrifugation, the supernatant that we will get, that will have our plasmid DNA. So after this neutralization, we will centrifuge it, and after centrifugation in the supernatant, we will have the plasmid DNA. Okay. Now coming to the next step of this plasmid isolation process, that is the purification of the DNA. Now we have collected the plasmid DNA in the solution. Now this supernatant, from this supernatant we have to isolate the DNA. For that process we use a column. So these columns are manufactured by companies, and these columns, they have a membrane which is a silica membrane. So this supernatant that will have this plasmid DNA, this supernatant is now loaded onto the mini column containing a silica membrane. So this column has the silica membrane here, and the supernatant is loaded onto here. The supernatant is loaded here. Now we will maintain high ionic strength in this condition so that DNA can bind to this membrane. Then the column will be washed by wash buffer, which will have a high ethanol concentration, so approximately 70% ethanol. With that we will wash the column. So this is a process of purification of the DNA, so that all the debris, everything can be removed, and the DNA can, the purity of the DNA can be increased. So after this washing step, then we will elute out this plasmid DNA from this silica membrane with either T buffer, so T is tris buffer, or water, right? So with T or water we will elute out the plasmid DNA. So what we will do for elution, so suppose we want to elute out our plasmid DNA in a very small volume to maintain a good concentration of the plasmid DNA. So suppose we give 30 microliter of water on this membrane, and then we centrifuge this column. Then after centrifugation, the solution from this column will come out here in this tube, and it will have our plasmid DNA. So that is how we will purify our plasmid DNA, starting from bacterial culture to finally purification of the DNA by using this silica membrane, by using this process called alkaline lysis. So after we purify our DNA, how do we measure the quality of the protein, or how do we measure the concentration of the protein, and how do we comment about its purity? For that, we have two methods. One is agarose gel electrophoresis, another is spectrophotometry. So this is a agarose gel electrophoresis of actually a digestion process. So this is how the this is how the image, gel image looks like. This is actually the 1 kb ladder. So this is a DNA ladder, right? Now this second lane, in the second lane there is this uncut plasmid. So the plasmid is done in this lane, right? And this particular band that you can see here, this represents the supercoiled form of the plasmid. What is supercoiled form? So when since this plasmid DNA is circular DNA, it forms this coiling, negative and positive supercoiling, and this supercoiled DNA runs faster in the agarose gel than the linear plasmid DNA. So actually the plasmid can exist in three forms. One is supercoiled, which is actually circular and forms supercoiling. Next is linear. Another is nicked. So in nicked DNA, basically there is one cut. So the circle is not complete. The circle is not complete in two strands. So in the supercoiled DNA actually runs fastest in the agarose gel. After the supercoiled, so the linear DNA, the supercoiled DNA runs faster in the plasmid, in the agarose gel, and on top of this supercoiled DNA we can see a band here, another band here, right? These are also the other forms of the plasmid. So this one is the linear form, and the nicked form is actually the most the conformation of this nicked form is not suitable enough to run fast in the agarose gel, which is why nicked form runs slowest in the agarose gel, right? So this is the supercoiled, the linear, and the nicked forms of the plasmid. So when we run the plasmid in the agarose gel, we will be able to see these three bands. But if our plasmid preparation is not good, we may see several bands, we may see contamination of other bands. So by that we can qualitatively ascertain the purity of the plasmid DNA that we have isolated by running it on the agarose gel. And the other method for quantitatively determining the concentration of the plasmid and the purity of the plasmid, we can use spectrophotometry. So what we do is we plot the absorbance, or we measure the absorbance of the nucleic acid. We know that nucleic acids, they absorb at 260 nanometer, right? So at 260 nanometer we can see this kind of a peak, right, at 260 nanometer. Now when we quantify DNA in a spectrophotometer, there are three wavelengths. There are the absorbance on three different wavelengths are actually taken into consideration. One is 260, another is 280, another is 230 nanometer. So we know that in 280 nanometer proteins absorb, in 260 nanometer DNA or RNA, and in 230 nanometer phenol and some other salts can actually absorb. So phenol is often used in the purification process of the DNA. So therefore if our protein contains phenol contamination, so if our DNA contains phenol conformation, we can have an idea about it from the ratio of the absorbances at 260 and 230 nanometer of wavelength. So now if our DNA has protein contamination, so suppose our purification process was not good, so there are some proteins in the DNA solution, so that we can have an idea from the 260 by 280 ratio. So if it is 1.8, it means that our DNA sample is pure, which means it has very less protein contamination. 260 by 280 should be 1.8, and 260 by 230 should be approximately 2 for a pure DNA. So from this vectrophotometric analysis, we can comment on the purity of the DNA from the 260 by 280 and 260 by 230 ratios, and also from the absorbance we can calculate the concentration of the DNA as well. So there is a standard which is actually taken into consideration here. So suppose if the absorbance at 260 nanometer is 1, it is considered as 50 nanogram per microliter of DNA. So 50 nanogram per microliter of DNA gives us this OD value of 1 at 260 nanometer. So from that we can calculate. So suppose if suppose if the absorbance value at 260 comes as 0.5, from this we can calculate what will be the concentration of our DNA. So suppose it will be 25 nanogram per microliter of DNA, right? So from the absorbance value we can directly calculate the concentration of the DNA, right? So that is how we can quantify the plasmid DNA that we have isolated. Any doubts so far? Okay. Alright. So in the next topic we will move on to the isolation of genomic DNA part. Now, so far we were talking about the isolation of plasmid DNA. Now, if we have to isolate genomic DNA is much larger. I already talked about this, that it is approximately 5 megabases, 5 into 10 to the power 6 bases in length. And for genomic DNA isolation also the process is somewhat similar. So first of all we have to inoculate a bacterial culture for overnight in LB medium at 37 degrees centigrade, right? In the next morning we have to collect the cell pellet, and then similar to plasmid DNA isolation, we have to first resuspend the pellet in lysis buffer and incubate at 37 degrees centigrade for 1 hour. We have to keep it for some time for lysis to complete. Then what we will do, this is actually Yes, Tanya, no issues. These all videos are available in the YouTube, so I will be uploading today itself after the session, so you can get an access to that, and that will actually be shared in the course website, right? In the NPTEL website, in the problem solving session, you will if you click at this problem solving session tab, it will give you link to a Excel sheet where you will get all these YouTube links from the problem solving sessions. Okay, so now for genomic DNA isolation, we have to add an equal volume of phenol chloroform. So this is a phenol chloroform method. So phenol chloroform we will add, and then we will centrifuge at very high speed at room temperature. So why do we add phenol chloroform? So phenol chloroform will actually help in phase separation. So what we will see after this centrifugation is that there will be an aqueous phase and an organic phase formed. So in the tube, in the upper part you will see an aqueous phase, and in the lower part you will see an organic phase. Because phenol and chloroform, they will separate the phase, right? So after adding equal volume of phenol chloroform, we will have to mix the solution very well, and then we have to centrifuge it at very high speed. And after that, we will see that there will be two phases which will form in the tube. The aqueous phase will mostly contain DNA. At the interface of these two phases, we will have the proteins, and the lower organic phase will have the lipids. Because lipids are soluble in the organic phase, right? So which is why lipids are hydrophobic. So lipids will be in the organic phase. At the interface, the proteins are both hydrophilic and hydrophobic, so the proteins will form the interface. And in the aqueous phase, DNA will be present because DNA is hydrophilic, right? This is how the phase separation will occur with the addition of phenol chloroform. And now, since the DNA will be present in this aqueous phase, so we will collect this aqueous phase very carefully so that we don't mix up these phases. And also we should not touch this interface area, because if we touch the interface area, we will collect DNA along with the proteins. So protein contamination will come in our DNA. So we have to carefully isolate this aqueous phase from the tube. So we remove this aqueous phase, and then we precipitate the DNA with ethanol. So approximately 2.5 to 3 volumes of ethanol we will add. And suppose, so the aqueous phase is 200 microliter. So 3 volume of ethanol will mean that 600 microliter of ethanol. 600 microliter of ethanol we will take. And then that will help in precipitation of the DNA, because DNA will precipitate in ethanol, and we will have to incubate it at minus 20 degrees centigrade for 1 hour. So after this incubation, we will have to centrifuge it at a very high speed at 4 degrees centigrade. Then after this centrifugation, the supernatant we can discard, because the DNA will now form the pellet. Now, so this pellet formation of DNA is done with 100% ethanol, right? Now next we will wash the DNA pellet with 70% ethanol. So for washing the DNA, we will always use 70% ethanol to rinse the DNA pellet. Then we will again centrifuge at very high speed, and we will again discard the supernatant, and then again the pellet will form. The pellet will have the DNA, right? Then what we will do is we will air dry the pellet. So we will keep it open, the tubes, we will keep it open for some time, so that the DNA pellets can be dried up totally, and once the pellets are dried, we can then again resuspend the pellet in TE buffer or in water. So that is how, with the help of this phenol chloroform phase separation, we will isolate the genomic DNA of bacteria. So far we have discussed about the structure of nucleotides, structure of DNA, and how we can isolate DNA, either the plasmid DNA or genomic DNA. Now this genomic DNA, not necessarily we have to isolate the genomic DNA of bacteria. We can isolate the genomic DNA of animal cells like the liver cells, kidney cells, or we can isolate the genomic DNA from the plant cells as well. But as I mentioned, that the plant cells will have these polysaccharides and will also have these secondary metabolites, which make the purification process a little bit more difficult. So that's all for today. Now let's discuss the question answers. So this is the first question. Please let me know your answers in the chat box. Yes, so to lyse the bacterial cell wall, we use the reagent lysozyme. Correct answer. Next question. Right. So from plant sources, the presence of polysaccharide and secondary metabolites affect the purification of DNA process. Next question. Yes, Tanya, tell me your doubt. Metabolism. Okay. So can I come back? To your question later? Just give me a few minutes. Let me discuss these question answers first, and then I will discuss your question. Yeah. Okay. So do you want to answer this question? Question number 3. Okay, right, that is the correct answer. The function of EDTA is all of the above. Okay, next question. Okay, let's see what is the answer. A and B. You're correct, Tanya. So the denaturation of genomic and plasmid DNA, as well as breaking down the cell wall. That is the function of NaOH. Next question. Okay. Amardev, do you want to answer? Alright, so the correct answer is that of Tanya's. So B is the correct answer. If RNase A is not added, then RNA will not be removed. DNA and RNA does not form any complex. So no, that does not happen. So there is not a complex formation that occurs between DNA and RNA. So that is why this is not a correct. This is not a correct statement also. So RNA will actually, since they absorb at same wavelength, so therefore the quantitation of this DNA sample then will actually be hindered because of the contamination of the RNA. No, actually DNA and RNA, this DNA RNA hybrid formation is energetically not very favored, which is why DNA will precipitate separately, RNA will also precipitate, but DNA RNA complex formation that does not occur. No, actually, the thing is, cytosine of DNA and guanosine of RNA, so that is correct. They can actually form bonds, right? They can form hydrogen bonds, but in this process, if RNA is present, they will exist in the solution. They will not form complex together. That is not something that happens. Not usually happens. So in the cell also DNA and RNA coexist. But does RNA always form complex with the DNA? That does not happen, right? If RNase is present, then it will chop off the RNA. So RNA will be removed. So all RNA molecules will be chopped off, cleaved. So RNA will not be present in the solution anymore, right? But if RNase is not added, then RNA contamination can be observed. And in the agarose gel electrophoresis, if you run now, you will see that RNA is much smaller in size, right? So RNA will form a band, or sorry, not small in size actually, than the plasmid DNA. RNA will be much bigger in size. So RNA will form bands. RNA will form bands up on the agarose gel. So you can see the plasmid band as well as the RNA band, and that will actually show you that your DNA sample contains RNA, because that you will not be able to determine from the spectrophotometric analysis, because from the spectrophotometric analysis both DNA and RNA will absorb at 260 nanometer, but when you run on the gel you can see the RNA band separately than the plasmid band. What you are saying, that one pair of base will be available of RNA. No, actually, if the bases are clipped off, or not the bases, the nucleotides, if the nucleotide, the phosphodiester bond, if that is broken, then they will not be able to form bonds with DNA strand anymore, because they will be single nucleotides, right? So which is why there will not be any such complex formation. Is that clear now? Okay, all right. Now moving on to the next question. Yes. So that's the correct answer. The rule of SDS is, what is DNA protein complexes? So basically, DNA protein complex means some proteins can bind to DNA. Okay, so if this kind of DNA, if we are trying to isolate, then these proteins which can bind to DNA, they can remain bound to the DNA molecule and the protein contamination can come. Or suppose when we are purifying proteins also, so this problem happens in both DNA isolation as well as protein isolation. So when we try to purify a protein, suppose the protein is a DNA binding protein, right? So there are several types of DNA binding proteins. Do you know, like for example, transcription factors? So these transcription factors are proteins which help in transcription, so they bind to the DNA. There are other molecules also, like for example helicases, they bind to DNA. So when these protein molecules, they have an affinity towards DNA, they can bind to DNA, they can remain bound to the DNA, and it is difficult to isolate DNA and protein in that case. So what we can do in that case, if we yes, this happens in cells also, yes, because for example, how does the transcription process occur? Transcription means copying of DNA information into the RNA information, right? So in this process, there are proteins like these transcription factors, or for example in the replication process, yes, I'm coming to your question. So for example in the replication process, there are proteins which bind to the DNA which has to be replicated, right? The polymerase, for example, the DNA polymerase, the helicase which unwinds DNA. So there are several types of proteins which bind to DNA, right? As well as histones. Histones are also proteins which bind to DNA. So they can bind to DNA, and yes, they can also be dissociated from DNA. But suppose you are trying to purify a polymerase, a DNA polymerase. Now when you grow the protein inside the cell, or when you overexpress the protein inside the cells, then inside the cells there will be DNA also. So these polymerase proteins which will be produced inside the cell, they will bind to the DNA. Now once you try to isolate the protein, for example, then what will happen? This particular protein molecule will remain bound to the DNA. So there are several things that can be done to actually isolate the protein and DNA. For example, we can treat it with DNase. Okay, DNase is an enzyme which can cleave the the phosphodiester bond between the nucleotides, right? So we can use DNase to chop off the DNA, right? So there are several other treatments also which we can perform for isolating the protein and DNA. And in case of DNA isolation also, so we have to carefully monitor that we should not take the protein interface. We should not touch the protein interface in case of phenylchloroform isolation, or in case of the plasmid isolation also, by alkaline lysis method, we will have to use the denaturing agents which will denature the proteins, so that now the proteins will not be able to bind to DNA anymore, right? We are using NaOH that will denature the proteins. We are using SDS which will denature the protein. So if the protein is denatured, the protein will become linear, right? So once it is linear, it will lose its affinity to bind to DNA. It does not have the conformation anymore to bind to DNA, which is why if we denature the proteins during DNA isolation, then we will get a DNA sample which will be free of proteins. Is that more or less right? Clear to you? All right. Okay. So yes, you're most welcome. So now moving on to the next question. Yes. So what is the purpose of plasmid isolation? Cloning, gene expression, vehicle of delegating target DNA in another organism? All of the above is correct. Okay. Next question. Yes, C is correct, but there is another answer. Another option is also correct. Yes. Yes, yes. So A and C, they are correct. Plasmid and genomic DNA, they are different in conformation because plasmid is circular. It forms supercoiled form, but genomic DNA is usually linear. And size difference, plasmid is much smaller than the genomic DNA. So genomic DNA is in megabases, plasmid DNA is in kilobases. Next question. Yes, the neutralization buffer contains potassium or sodium acetate. Next question. Okay, so here the correct answer is all of the above. Alright, next question. Yes, the correct answer here is DNA can be eluted by the addition of water. Next question. Yes, ethanol or isopropanol is the correct answer. Next. Correct. B and D. Both jelly electrophoresis, agarose jelly electrophoresis, and the UV spectrophotometric analysis. Next question. Yes, that is the correct answer, 260 nanometer. And the last question. Yes, so the correct answer is that plasmids are supercoiled, so they migrate faster than the linear DNA. Alright, so that's all for today. Now coming back to your question, Tanya, what is metabolism? So basically metabolism is all the reactions, the biochemical reactions that occur in our body. So what kind of biochemical reactions? For example, the some very common examples I'm giving. For example, the food that you intake, this food is broken down to form very simple molecules, right? So from carbohydrates are broken down to produce the monosaccharides like glucose. Proteins, they are broken down to produce amino acids. The lipid molecules, they are broken down to produce fatty acids and glycerol, right? So this process of breakdown is metabolism, right? The biochemical reaction that sustain life, right? That is metabolism. Now all these reactions that I talked about, they are breakdown reactions. Now metabolism is not only about breakdown, metabolism is also about synthesis. So once the glucose has broken down, from this glucose will be first of all utilized in our body by the process of respiration. So if you know respiration is the process by which glucose can be utilized to produce energy, right? So that is again a process of metabolism, again a part of metabolism where glucose is being utilized. Next is glucose can now be converted to other higher order macromolecules also. So glucose, if it needs to be stored in our body, it will be converted to glycogen. So glycogen is a compound which is stored in our muscles, for example, right? So this production of glycogen from glucose is again a process of metabolism, and again a metabolic activity. But this is a synthesis process. From amino acids, new proteins will be synthesized in our body. That is also a process of metabolism. That is also a synthesis process. From nucleotides, DNA will be produced. From fatty acids and glycerol, fats will be produced, or the lipids will be produced, right? So these are all synthetic processes. So they are like synthesis, they perform synthesis of one particular compound. This is also metabolism. So metabolism is usually divided into two segments. One is a catabolism, another is an anabolism. Catabolism is when the components are broken down to form simpler compounds, right? So here usually what happens, energy is produced in this process, right? So the bigger macromolecules, like for example carbohydrates, they are not being broken down to glucose, right? They are broken down to form simpler molecules. That is catabolism. And anabolism is the synthesis part, when glucose is being converted to form glycogen, right? So this is all about metabolism, the biochemical reactions that occurs in our body, inside our body, and can be divided into two types of reactions. One is catabolism, another is anabolism. So is that more or less okay, Tanya, to give a very brief introduction about metabolism?
Yes, ma'am. Thank you so much.
Okay, all right. So that's all for today. Thank you for joining. We will meet again next week And for the lectures, please go to my YouTube channel. And thank you so much. We will see again in the next week.
Ma'am, I have one doubt if you don't mind.
Okay, no, sure. Please tell me.
Uh, Ma'am, it is regarding not one doubt. I have actually a few doubts in chapter water of biochemistry.
Okay.
If I can ask you.
Yeah, sure.
So can I share my screen?
Okay, let me unshare my screen first. Yeah, now you can.
Okay, ma'am. Ma'am, is anything visible?
No, you are sharing, but you have to go to the screen.
Okay. Ma'am, is it now visible?
Yeah, it is visible.
Okay. Ma'am, if you see here, no, that here it is written that water have very less tendency to ionize. Then, ma'am, how we can write H plus OH minus?
Oh,
If
yeah.
like,
So actually, pure water does not ionize, right? So H
okay,
plus and OH minus, this thing that we write like with much ease, does not occur with such ease, right? So what we have to do is that we have to add little bit of acid to
okay,
actually ionize water.
okay. So, ma'am, then here it is written that even if nothing is added, pure water is like H plus and OH minus, and this much is present already. Then, ma'am, how it is present? Like, if it is present, then how we are able to drink the water? Because, like,
No,
I don't
actually,
want
it
to.
states that in the pure water, it contains
Mhm.
equimolar amounts of H plus and OH minus, but does not mean it remains in an ionized state.
Okay,
Right? It is ionized once you add a little bit of acid, then this ionization process becomes faster, right? But the
okay,
thing is, it does not. It just means that it contains equimolar amounts of these ions. Means that that is present in pure water, but it's not in an ionized form. But in like in the respect of molarity, how much moles of H plus and OH minus is present, that is what is stated here. So the
okay.
pH and ionic product of water, those things are calculated here, right?
Right, right.
Right, yes.
So water is present as H2 only, not H plus and OH minus.
Not pure water, but um, so do you understand the difference between pure water and water where there are some other ions present?
Right.
Right. So if it is 100% pure water, that means does not contain any other contaminants, then it does not dissociate into H plus and OH minus. Like that is what is stated here.
Oh,
Ideal condition.
okay. So the water we drink that is not pure. So in that water it is H plus and OH
Some
minus.
amount of H2O is always dissociated in that way. Yeah.
Okay, okay, ma'am.
But not
And
all.
ma'am,
Yeah.
okay, okay, ma'am.
Yeah.
Okay, thank you.
Okay.
And here they also showed this proton jumping, which is okay. I understood that, but they have also told that a similar thing happens with hydroxide ion, OH minus.
Hmm.
So, ma'am, I searched on internet a lot, but did not found this.
The proton transfer on the OH minus ion, right?
No, ma'am, this hydroxide ion jump. Here they are showing proton jumps, right? So this is very okay. This I understood.
Okay.
But they say that hydroxide ion also jumps in the same way. But when I searched on internet, I did not found any related thing.
Actually, you know what? This is what I assume that this is
Mm-hmm.
talking about the proton jump in the hydroxide ion actually. In OH minus
Proton.
ion, yes. So, uh, just today also I showed you one thing, that is the I don't think you were there. So I was talking about this DNA, how DNA is more stable than RNA. So there what happens is that there is a hydroxyl group in the RNA molecule, right, which can donate its proton. If it donates its proton, then it actually the phosphodiester bond between the two nucleotides actually break if it donates its electron, right? So that is how the hydroxide ion also can donate its electron or can accept the electron. If it accepts the electron, then it exists in a different state, right? So this kind of proton jump that is shown in H2O, right? This is a structure of H2O. So basically the hydrogen bond formation actually is so the change in the hydrogen bond formation takes place, right? So that also happens in case of OH minus ion also. So if there is OH minus ion or hydroxyl group present in the in any particular molecule, it can if there is, suppose you add base in the solution, then what will happen if there is a hydroxyl ion, the base will try to take out its proton, right?
Yes.
So then the OH minus will become O minus, right? So that is how the jump can actually take place.
Okay, ma'am, got it. And OH minus ion, like we can also add any acid to it, then this will happen, or
then
this
what
won't
will happen? What do you think?
Then H should be taken by OH minus.
Yes, exactly that.
Okay,
And that is what happens in RNA's mechanism. So in the previous week 8, if you go to my week 8 lecture, you will find the mechanism of RNase A, which we have discussed. And there it was shown that this RNase A, the active site of this enzyme, contain two histidine molecules. One is histidine 12, another is histidine 119. So one histidine molecule acts as a base, another acts as an acid. So which means one accepts proton, one donates proton. Right. So that is how it can actually break the phosphodiester linkage of a RNA chain. So if you go to that lecture, I think it will be more clear to you in this regard.
Okay, ma'am. Okay. Just one more thing.
Yeah, sure.
Okay. So, ma'am, this is the equation, no?
Henderson-Hasselbalch equation, yes.
Yes, ma'am. So it is written over here that this is not valid for strong acid and strong base. So I did not understood why they told like this. They have showed here something calculation, but I think
so Henderson-Hasselbalch equation is actually for weak acids. So what is the difference between strong acid and weak acid?
Like strong acid can dissociate completely and quickly, maybe. That's the difference.
Complete dissociation is observed in strong acid and strong bases. So this equation, that log of base by acid, right? So here if it is a complete dissociation, then you cannot put it in this equation. Think about it. The concentration of the conjugate base by acid.
Conjugate, yes. So, ma'am, we can put, no? In fact, okay, log 1 is 0? No, but how log 1?
No, not log 1. But think about it. So, when there is a reaction going on, so if at a particular time point, the acid is completely dissociated, then the denominator will become 0.
Okay, ma'am, got it. Fine.
Okay, because there will be no HA, or the acid will not be present anymore.
Okay, ma'am. But then, ma'am, pH will be equals to pK, no? So at least we got the value of pH.
But if there is 0 in the denominator, it becomes undefined.
Ma'am, this term became yes. Okay,
Which is
fine. Then we
why
can.
we cannot
Oh,
use this particular equation, Henderson-Hasselbalch, for strong acids and strong bases.
okay, ma'am. And ma'am, PK and pH always makes me confused. Like I
pH is the
always
negative
find
logarithm of the hydrogen ion concentration or the hydrogen ion activity, right?
right.
But pK is the dissociation constant of the acid or the base. Base is
Okay.
we can write pkb also, but it is the constant of dissociation. That means how much the acid dissociates,
Okay, ma'am.
right?
Okay. So how much it dissociates and how much it produces H plus, and that's why we are considering here
pH,
pH.
yes, exactly.
Oh, okay, okay, ma'am. Uh, there was this one graph. Uh, yeah. Yes, ma'am. I did not understood this sentence. This
uh, can
from
you
here.
can you read it out for me?
Yes, yes, ma'am. It says that the pH of blood in healthy individual is closely controlled at pH 7.4 plus minus 0.05. The phosphate and bicarbonate ion in most biological fluids are important buffering agents because they have pKs in this range. But we are talking about pH, and now they are
Okay,
keeping your pK.
okay, because in this biological fluid, for example in the blood, the pH has to be first of all very closely monitored. Why?
Right.
Because in the proteins in our body, the enzymes in our body, all of their function is dependent on the pH, right? If the pH changes, then their charges will also change, which will affect their function. So that has to be closely monitored, so it has to be very well controlled. That is the first thing.
Right.
Next thing is the how is this pH maintained? So there are phosphate ions and there are bicarbonate ions in the um, in our body, in our blood also there are these ions, phosphate and bicarbonate ions. So
Okay.
they have this pK in a close range to the blood pH or the pH of the biological fluid. So that their pK or the dissociation constant of these ions means the acids, the phosphoric acid or the carbonic acid, right? They
Right.
actually form these bicarbonate and phosphate ions, right? Upon dissociation. So their pK, since their pK is very close in this range, they have this pK in this range. So that is how their dissociation actually controls this pH of the body fluids or the blood. Do you understand?
Oh,
Since pH equals to pKa plus log of base by acid, so the
right.
dissociation of these kind of acids like the phosphoric acid, carbonic acid, they
Mm-hmm.
once they dissociate, they will form these ions like the phosphate ions, the bicarbonate ions, right? And
Right.
their pK, that is why the pK of these acids is important for maintaining the blood pH.
Okay, okay, ma'am. So they are weak acids.
They are weak acids, yes.
Okay, okay, ma'am. Uh, okay. I'm sorry, I am taking your time, but
No,
I
it's
don't
it's
know
fine.
about.
It's fine. Please ask me questions. It's always good to interact.
Okay, so ma'am, there was one another graph. So in that graph, only one value we were counting. Like
Yeah,
here, only one. But in
yeah.
this graph, there are two equivalent points. So can we count PK for this 2 point, or we cannot?
Actually, I had
Um,
discussed this in detail in week
okay,
2, I think. Um,
okay,
there was this discussion about the titration of different amino acids, and for
okay,
calculating the pK in this kind of graph, where there are 2 points of inflection. So what you can do is that you have to actually check the charge of the amino acid. So from one point to another, what is the charge of the amino acid on the amino acid? That is, in the zwitterionic form, it exists in zero charge, right? The net charge is zero.
Right.
Now the pK that you have to consider for calculating the pi of this kind of amino acids is that when the amino acid changes from is -1 to 0 and then from 0 to +1. So when the charge of the amino acid changes from -1 to 0, at that point 1 pK will be responsible for that. That pK you have to take. And when the amino acid, the charge of the amino acid changes from 0 to +1, there will be 1 pK which will be responsible for that change. Now these 2 pKs that you get in this zone from -1 to 0 to +1, these 2 pKs you have to select and you have to make an average of them. That will give you the pi value of this amino acid.
Okay, ma'am, -1 0, I did not understood that.
For that, actually, you have to check this lecture, because
Okay, ma'am.
when it is -1, so when there is COO minus in the amino acid.
Okay.
When is it +1? When there is NH3 plus on the N-terminal end of the amino acid, right?
Right.
When is it 0? When there are NH3 plus and COO minus together. That is a zwitterion.
Right.
And when there is any other charge, how is it possible? Because the side chains of the amino acid, they also contain some groups which can ionize, which can form ions, right? For example, in basic and acidic amino acids. So lysine, for example, in the side chain of lysine, there is another NH2 group which can form NH2 plus, right?
Okay.
Or NH3 plus. So then this charge will also be calculated, right?
Okay.
And this is a titration curve of which amino acid? Is it written here?
It says a titration of
A polyprotic acid. Yes. So
Yes.
basically, polyprotic acid means that there are several OH. Actually, this is an amino acid which contains several charges, right? So which is why there are these 2 inflection points you can see, and for calculating its pi, you have to consider only the +1 to 0 to -1 to 0 to +1. That point.
Okay.
So if you want to understand this in detail, please go back to this YouTube, my YouTube channel.
Okay.
I think in week 2 we had discussed about this.
Okay, ma'am. Ma'am, I also wanted to go in detail of this PI, PK and all those things. So
that is also there. That is also there in the week 1, I think.
Okay, okay, ma'am. Okay.
Henderson-Hasselbalch equation.
Okay, ma'am. Okay. I had doubt in this, but I will keep this in discussion forum because I think you will have to go through all this. So
all right.
I will.
There's a calculation. Okay. You put it in the forum.
That's why I will. Yeah, because it is not possible here. So one theoretical doubt I have. Just a second. Yes, here, ma'am, it says that delta G will. Okay, just a second. Yes, here this. They say that thermodynamic process favored because increase in entropy, which is correct because according to this equation, here is entropy.
Yes.
So does that mean that always delta G will be negative? Because
if delta G is negative, then it is thermodynamically favored. But not all reactions that take place in our body, for example, they do not always have negative delta G. So then how does they take place? They take place in so they are accompanied by one other reaction which has a very negative delta G value.
Okay.
So one reaction has a positive delta G. It is accompanied by another reaction which has a negative delta G. So
Okay.
in when they are when they occur together, the overall delta G becomes negative.
Okay, ma'am. Okay.
Right. So that is how actually that is why you have to you have to actually um, in various cases, like for example enzymes, several enzymes use ATP for several
Mm,
actions, right? So ATP, what
okay.
happens in ATP, so when ATP is broken down to form ADP, the
Yes.
release of the inorganic phosphate releases so there is a huge change in delta G. So the
Yes.
negative change, right? So delta G is
Yes.
very negative in that reaction when ATP is broken down to ADP and inorganic phosphate, right? So that is why
Okay.
many enzymes in our body are coupled with this ATP hydrolysis,
Okay. Okay, ma'am.
right?
So even if the another reaction which is happening is positive giving delta G, then also we consider the overall process and overall delta G
Overall delta
negative.
G change, yes, yes.
Okay, okay, ma'am.
That is
Okay,
why
got
they are
it.
coupled, and that is how they can uh, they can uh, they are performed in our body. Otherwise this obviously if the delta G is positive, then the reaction will not occur spontaneously, right?
Yes, ma'am.
Yes,
Okay, got it, ma'am. Thank you so much.
yes,
This
yes.
is this is my last doubt. Like,
Okay,
see, ma'am, non-polar. If there is anything, then they form because of the hydrophobic bonds only, right?
right.
The lipids and all this thing, hydrophobic bonds they
Yes,
form,
the
and
hydrophobic
then only
interaction.
all these things are possible. Then also they have written over here that it is inappropriate to say that non-polar substances are result of hydrophobic bond, but it is like that, no? So why?
Oh, I think they are, I think, trying to say that it's not a bond, but
Okay,
maybe that is what they are trying to say, that it's a
yes, something
hydroph.
like that. Maybe for us to think little more. That's why they have given this
Maybe,
sentence in the
maybe that
slide.
is how might be. It
Oh,
might be.
okay, ma'am. But in non-polar molecule, there are van der Waals forces, right?
Van der
Again,
Waals
that
forces is
had
there. Hydrophobic interaction is there. Like when two molecules, when they are hydrophobic, they come in close contact. Why they come in close contact? Because they want to avoid the other hydrophilic environment. So for
yes,
suppose it is
ma'am.
given to just like oil separates in water. So the
Yes,
molecules of oil, they form a separate layer above
yes,
water.
ma'am.
Why?
Yes.
Because they do not want to face the water, because they are hydrophobic. So that's how.
Yes, ma'am. But ma'am, in non-polar molecule also, if there is van der Waal forces, right, which is result of positive and negative electron cloud getting separated, right?
Mm-hmm.
So at least they have that positive and negative ion. So why they cannot form bond with water itself? Like now they already have positive and negative.
No, actually, bond formation, covalent bond formation, for example, it requires sharing of the electrons, right? That does not
No.
happen.
Yes, that covalent bond, it is okay if it
Yeah.
does not make, but at least it can make the hydrogen bond because hydrogen bond does not require any sharing.
For
So, like,
hydrogen bond, you have to have an electronegative atom.
yes,
If that is present, then it can form a hydrogen bond. But if the electronegative atom is not present, so for example oxygen or nitrogen, which can share its hydrogen with another compound, right, another molecule, so if that is not present, then there will be no such hydrogen bond formation, right?
okay, so
What
which
happens,
means
for
that,
example, yeah, sorry.
okay, so that means that the delta positive charge, which is on hydrogen, when combined with oxygen, that delta positive charge is much more than the positive charge created by the van der Waal
Exactly,
forces.
exactly, exactly, much more
Okay,
in
okay, okay, ma'am,
strength.
got it. Okay, that's it. Thank you so much, ma'am. I
Okay.
was very confused with all this thing. This is actually what you will be knowing that.
Yeah, yeah,
So
I
I,
understood.
yeah, so I
So
marked.
the thing is, you can also consider another book which is Leninger. Sometimes
Okay, ma'am.
Void is a little bit more difficult in a first read. Okay,
Okay.
so Leninger is a book which is a little bit more easier in terms of like the when you're reading for it for the first time, right? So you can consider reading from Leninger. Voigt is also a very good one, but the language is a little bit difficult here and there, and things are not very easily explained as that of Leninger.
Okay, ma'am.
Right,
Sure.
and if you have any doubt regarding the lecture materials, regarding whatever is taught in this experimental biochemistry, you can always join my sessions, as well as you can go back to the previous sessions and look them up in YouTube. They're all available, right?
Thank you so much, ma'am. I was much worried with this subject, so
don't worry, this is a subject to love, not to worry about.
okay,
If you
ma'am.
understand it well, it's a very, very interesting subject.
Okay, ma'am. Thank you so much for your time.
Okay, you're welcome, most welcome. Okay, bye then.
Okay, ma'am. Bye.
Okay, see you next week.
Yeah, ma'am.